All right, so again, we're going through genetics, and we're going to start talking about things like dominant recessive genes and such. And so we left off on this Punnett square Punnett square-like chart on genotypes and phenotypes of different types of blood. And again, the type of blood that you have as a student is a mix between your mother and your father's blood types, and whatever they pass down is the blood type that you got. All right, so again, just to reiterate some important things, you get half of your genome from each of your parents. And so those two halves come together and then make your genome. And again, this is called hereditary, which is the passing down of genetic information. All right, genotype and phenotype, we should hopefully know those. All right, hopefully we don't have any mutated genes. And again, a lot of the bad genes, if you have two recessive alleles passed down, one from mom, one from dad, that recessive trait will show up, and those tend to be our kind of disorders and such. All right, this was our stop sign, and then this is our new slide. All right, so just a couple different types of recessive disorders. In the left column, you'll see each of those things. So PKU, cystic fibrosis, Tay-Sachs disease, and such. In the middle is all of their descriptions of each disease. Definitely don't want any of these. And over here it just says that they're all recessive, and most of them are autosomal, which means that they are actual effects on your body, not like something like a sex-linked gene. We'll get to those soon. All right, so just moving on from here. All right, this is just a little bit of a breakdown. All right, so at the very top of this chart is the parents. All right, so circles are dictating females, squares are dictating males. All right, so up here is parent generation 1. All right, each time we go down a level following this line, this is their offspring here. All right, so mom and dad had a little girl, and then that girl got married. All right. And this married couple here had 3 kids that you can see as the 2 circles and the square. And anything that's filled in is showing a offspring that has the trait. So this is just kind of showing how one parent back in the first generation can produce offspring that are carriers. All right, they're not showing up with these traits, but once they have offspring themselves, they're still carrying that trait, and they can still pass that down. All right, so this is just a little bit of a diagram showing on how you can pass down things hereditarily. All right, so just recap, a dominant trait can be identified because many of the people in the same family will have that same trait. So typically, if you have brown hair, most of your family members will also have brown hair. But every now and then, a brown-haired woman and a brown-haired male can procreate, and then they can produce a blonde child. Someone who is blonde is most likely double recessive. And typically, if you see 2 blonde parents, you will also see a blonde offspring, most often. All right. And so dominant traits, free ears, so that means that they're detached. Tongue rolling, so that means physically rolling your tongue into like a tube like that. That's genetic. Some people just can't do that. Freckles, and then polydactyly, that's just having extra fingers or toes. Hopefully you don't have an extra finger or toe. All right. Recessive traits, we saw those in those charts before, but they can be identified in a family because only a few are affected. By 2 normal parents can produce a child with that trait. So again, those brown-haired families, most people are going to have brown hair, but every now and again, you're going to see a blondie show up, and that blonde hair is a recessive trait, still carried by most of the family, but not expressed, is how we say it in the biology vocab. All right. So some examples of that are cystic fibrosis or Tay-Sachs disease. Again, recessive diseases and just dysfunctions typically don't show up. But if they do, they're unfortunate, but they're there. All right. So this is what we're getting to next. These are sex-linked or X-linked traits. They can be identified in a family if more males than females have that trait. And so this is kind of getting into those codings. Males are XY, females are XX, and we're going to see that actually more X-linked traits are carried on the X chromosome, and that's just because it's bigger. Examples of this are color blindness, hemophilia, which is a problem with your blood. Prefix hemo means blood, and then muscular dystrophy. So just going into this next slide, X-linked traits, genes found in the X or Y chromosomes are said to be X-linked. So most of the genes are found on the X chromosome because, again, it's bigger. And males, we only have one X chromosome. Therefore, all X-linked alleles show up in males, even if they are recessive. Because they're on the X and males only have one X chromosome, there's not another X chromosome to kind of trump that first one. The Y chromosome is obviously different than the X, and that's why even if those traits are recessive, they'll still show up in males. All right, so moving on. Examples of X-linked traits, again, color blindness, hemophilia, and then muscular dystrophy. All right, a sex-linked problem. So a cross between a woman who is a carrier, which is heterozygous, that means that she has two different alleles for a trait for color blindness, and a man without color blindness who has normal vision. All right, and so this X big C, X little c, all right, times X big C Y, all right? We're going to see that cross here. So this is the woman's alleles up on the top, and this is the male's allele on the bottom, or the side, sorry. And then try to fill this out on your own, but just to think of the top left square, what's going to go in there, all right? The mom's going to pass down that X big C, and so is the father. It's X big C. Right, and then you just carry those alleles down and those alleles across for the mother and father, respectively. And then once you have your combinations, right, you will see that, unfortunately, this bottom right corner is X little c Y, and that is the colorblind boy, right? So just a little bit on that. And then non-disjunction, all right? So this is actually a double negative, all right? So this is actually getting towards chromosomes and alleles separating from each other. So they're supposed to separate into two. So looking at this first leftmost flow chart, you have two chromosomes up in that top circle, and then they're going to split off into two separate bodies, a chromosome apiece. All right, that's the second tier. And the bottom tier, you're going to get 4 daughter cells, each haploid. All right, so each is going to have 1 chromatid per daughter cell. That's what's supposed to happen. All right, but sometimes this doesn't go correctly. All right, so look at the middle one. All right, you have 2 chromosomes in that top one. All right, in the second level, it's supposed to separate into 2 separate bodies, a chromosome apiece. All right, but you can see that both chromosomes actually went to that leftmost body. Right, and so when you look at the 4 daughter cells produced in the middle, 2 of them have no genetic information in them. That's bad. And then the other 2 still have too much in it. All right, and we learned that too much DNA is also bad. All right, and in the rightmost diagram, again, we're just seeing how this can go wrong. 2 chromosomes up top splits down into 2 separate bodies correctly, and then the bottom, the 4 daughter cells, 2 of them are correct. They have a haploid number of chromosomes, and then in the other 2, one of them has none, and then the other one has too much in it still. All right, so non-disjunction, so a double negative. A junction is the meeting up of 2 things. A disjunction would be the splitting up of those 2 things, but a non-disjunction means they never split up at all. All right. Double negatives get confusing, but hopefully you can follow that. And I believe this next slide is our last slide. Right? Non-disjunction results in either too much or too little. Right? You don't want gametes being produced with an incorrect amount of genetic information. We know that we need to have a total number of 46 chromosomes once mom and dad's genetic information combines. Right? And so we can see that too much, all right? We talked about trisomy 21, all right? That's Down syndrome. Turner syndrome, monosomy, all right? And then also Klinefelter syndrome, all right? That's an XXY. So this is a too many sex chromosomes here, right? XX, we know that that is a female, but XXY, this is going to be someone who has both male and female parts, right? You don't want that. All right, so this is going to be the end. All right, so I'm going to stop sharing my screen, right? And then following this video, I would like you all to complete the Word document I'm posting with this with just some vocab on it. Most of the vocab was going to be found in that glossary on our Google Classroom page, and some of it may just have to be a Google search. All right. Thank you for all of you who watched yesterday's video and then did the face lab. I got a lot of submissions yesterday, so thank you for that. And just keep up with the work, guys.